Today we are going to see the PCR. PCR polymerase chain reaction. आज हम देखने जा रहे हैं PCR टेक्निक पॉलीमरेज चेन रिएक्शन. क्या होता है मतलब पॉलीमरेज चेन रिएक्शन का मतलब क्या होता है? A number of copies are produced from a single gene in a vitro condition. In vitro condition means out of that cell. When the number of copies are produced within that cell, then it is called the vivo condition. And when it is produced in out of that cell, then it is called the vitro condition. So the number of copies are produced from the desired gene, whatever may be that desired gene, we can produce the number of copies with the help of that polymerase chain reaction. In that polymerase chain reaction, in that polymerase chain reaction, number of copies are produced in a vitro condition. In a vitro condition, that is artificially number of copies are produced. We have seen that in a recombinant DNA technology, the number of copies are produced when the gene, such a desired gene, is introduced in a cell, in a what we call it is in a transforming cell, and the number of copies are produced, but it requires a time. But whenever we want the number of copies within a stipulated period, then here the PCR machine it is used, or PCR technique is used. So this is the thermal cycler. With the help of that thermal cycler, we can produce the number of copies, and it is a device. With the help of that, we can produce. It is a device which is used for the gene cloning. It is produced billions of copies. It produces the billions of copies of a desired segment of DNA or the RNA. So with the help of that machine, we can produce. Which are the different requirement? The desired segment is the first requirement. That is the desired segment. Which is the gene, which is number of copies which are produced from that gene. This segment is called the desired segment. It may be for insulin, it may be for the follicle stimulating hormone, it may be from that endangered plants. Nowadays, the endangered plants are there, and they are now in a vanishing condition. So such genes can be collected from that plant, and it can be multiplied with the help of the PCR technique artificially. Then the deoxyribonucleotide triphosphate molecules we required here, because these ribonucleotide triphosphate molecules, they are complementary to the plant or the endangered plant, as well as the primer molecules are needed. The primer molecules, there are the two types of the primer molecule, forward primer molecule as well as the backward primer molecule. The forward and they are made up of oligonucleotides. You know that the primer molecules are needed because here it attracts the complementary nucleotides. We have seen in the molecular basis of molecular basis of the inheritance, or the genetic basis of inheritance. During the new strands are produced on the single stranded DNA during that replication of DNA. So the primer, it attracts the nucleotides, complementary nucleotide. Then the heat stable DNA polymerase enzymes. DNA polymerase enzyme is required to produce the new strands present on a template strand. But it is a heat stable, and it is generally produced from the Taq polymerase enzyme. Taq polymerase, that is Thermus aqueous bacterium. From that Thermus aqueous bacterium, this Taq polymerase enzyme is synthesized. Then the Mg++ ions are required to cut the DNA. So what is the mechanism of this PCR technique? First of all, here the DNA sample, the DNA which is to be amplified, it is collected. Then the primers, they are also added. The nucleotides, the adenine, guanine, cytosine, and the thymines, these are the different nucleotide, and this is the Taq polymerase enzyme. As well as here the buffer solution is here. This buffer solution is also added. So the mixture of DNA segment, desired DNA, mixture of that primer, nucleotide, stack polymerase enzyme and the buffer solution. This mixture, it is kept in the ependrum tube. And this in this ependrum tube, it is kept in that thermal cycler or the in this machine. Then what happens? Which are the different steps for the PCR technique? This is the first. This is the desired gene, which is to be cloned or which is to be amplified. Number of copies are produced from that gene. So this desired gene, it is mixed in that above solution. Then at a 97 degrees Celsius, in a new syllabus, it is given that 90 to 98 degrees Celsius. At a 90 to 98 degrees Celsius, this denaturation takes place. The denaturation means here the single stranded DNAs, they are produced from the double stranded DNA. You know that the complementary genes are present or the nucleotides are present on the gene. A pairs with T, G pairs with C, C pairs with G, as well as the T pairs with G. So here these nucleotides, they are joined together with the help of weak hydrogen bonds. So it has to firstly, that weak hydrogen bonds, it has to break firstly. And due to the breaking of that, here these two single strands, they get separate from each other. And this is called the denaturation. And it takes place at a 97 degrees Celsius. Then the second step is the annealing. In that, here you know that the forward RNA primer and the backward RNA primer we collected here, and the joining of that primer and the forward and the backward RNA primers on a single stranded DNA is called the annealing. And this steps, it takes place at a 55 degrees Celsius. In a new syllabus, it is given as a 40 to 60 degrees Celsius. So the joining of the RNA primer on a single stranded DNA is called the annealing. Then the extension, the third step is the extension. In that, the RNA primer, it attracts the complementary nucleotide. And the forward chain as well as the backward chain, it is produced on the single stranded DNA. So this is the extension step, and it requires the 72 degrees Celsius. So there are the three steps. The first is the denaturation, second is the annealing, third is the extension. In denaturation, the weak hydrogen bonds get break, and more amount of energy is required to break that GC bond between the GC bond, because here the three weak hydrogen bonds are present in between that G and C instead of the besides that of that A and P. Then annealing, in that annealing, the RNA primers are added on that single stranded DNA. And once the cycle, one cycle is required to 3 to 4 minutes. In automatic thermal cycler, the above 3 cycles are automatically repeated 20 to 30 times. And thus at the end, the n of the cycles, that is twice n copies of the produced. Once the desired cycle is completed, the amplified DNA segment is purified in a gel electrophoresis. After the sequencing segment is inserted into a vector and it is kept in a library. So as like this, here the number of copies, the billions of the copies are produced with the help of that three steps. Then what are the applications of that PCR technique? Once the desired, or when we select the gene which is to be amplified, then this gene, the number of copies or the multiple copies we can produce artificially within a short period, within 2 to 3 minutes, the 2 copies are produced. That is within that 20 to 30 minutes, we can produce the billions of the copies. And it is used in a to DNA library or the gene library, as well as for the cloning we can produce, as well as for the vaccination we can use this technique. So today's PCR technique, in that PCR technique, the number of copies we can produce artificially, because naturally when we want the number of copies from the cell, it requires more time. That's why we can produce the number of copies of that desired gene artificially in a vitro condition. Okay, thank you.